Hello and welcome to this webinar, Harnessing the Power of IgE Antibodies to Create Novel Therapeutics. I'm Megan Thomas, Multimedia Editor at DDW, and I'm delighted to say that today's webinar will be presented by IgGenix's Jessica Grossman and Derek Croote. IgGenix is taking a novel approach towards breaking through the last frontier of immunology, allergies. By re-engineering key antibodies in the allergic cascade, this biotech company is developing a first-in-class therapy for the millions of people whose daily lives are limited by allergies. In this webinar, you will learn about the use of native human IgE sequences as a foundation to develop optimal therapeutics, the benefits of single-cell RNA sequencing for understanding IgE and disease, the lessons learned in building a therapeutic pipeline and overcoming workflow barriers, and how the right technology can accelerate workflows while maintaining the agility required for a startup. Before we start, some background on Jessica and Derek. Jessica Grossman joined IgGenix in November 2020 as the CEO, bringing more than 20 years of healthcare leadership experience to the company. Most recently, she served as CEO of Medicines 360, a global non-profit drug company with a mission to expand access to essential medicines for all women. Jessica serves on the board of directors of Dare Biosciences. She holds 11 issued patents and has published several peer-reviewed articles. She earned her MD degree at Thomas Jefferson University and her BAs in Biology and American Literature from Brandeis University. As co-founder of IgGenix, Derek Croote is developing single-cell RNA sequencing technologies that revolutionize our understanding of IgE biology. Derek is the lead author of a publication in the journal Science that describes the first characterization of single human IgE B cells and the allergen-specific antibodies they produce. He received his BS from Brown University and PhD from Stanford University, where he was a National Science Foundation and Stanford Graduate Fellow. Now over to Jessica and Derek for their presentation.
Hello, thank you for joining us. If you're where we are over on the west coast of the US, it's the morning, so good morning. And if you're over in Europe, it's the evening, so grab a glass of wine and listen to us talk about harnessing the power of IgE antibodies to create novel therapeutics. So first I'm going to start with the introduction, and thank you so much for that lovely introduction that we just received. This is quickly just myself and Derek, who's right here with me. As was just noted, I've been a serial entrepreneur working in healthcare for the last 20 years. And Derek is the co-founder of the company and the CTO and really the inventor of the technology. And, you know, we're delighted to be here and to tell you about what we're doing because we have a really impressive library of IgE sequences, and we're really excited to be here in partnership with Drug Discovery World and SBT LabTech, who has helped us with our automation, and we'll touch on that a little bit as well. So quickly about IgGenix, we're a preclinical venture-backed biotech company, and we use single-cell transcriptomics to discover the rare IgE-producing B cells directly from patients. And our goal is to create a therapeutic for passive immunization or desensitization. We are the world leader in IgE biology, having the largest library of IgE sequences. And IgE is implicated in a variety of diseases, but the low-hanging fruit is in food allergy. And this is a large unmet medical need. We believe we can truly change the face of allergic disease for the millions of people out there who are suffering every day with food allergy. We'll talk about our platform, which we call the Seq Sifter. Derek will go into detail on that. We'll also talk about how this is a novel therapeutic approach in terms of class switching the IgEs that cause disease into IgGs, but it's based on a mechanism of action that's been well characterized. And then, we're excited. We're a small but mighty team. We spun out of Stanford University. We have an exclusive license to a patent from Stanford, and we're just on the cusp of starting IND development on our lead product candidate, and we should be in the clinic in about a year or so's time. So just a quick word on food allergy and allergy in general. This is the last frontier in immunology, and many immune-related diseases have really been transformed by biologics like asthma, rheumatoid arthritis. For food allergy, there's only one FDA-approved product that's approved only in pediatrics for peanut allergy, and we believe that we can really change this market by offering biologics to folks that are specific, so they will have no broader immune modulation or potential deleterious effects on the immune system. And we can offer a specific antigen-specific based therapy. There are 32 million people in the US who have allergies. It's a huge problem that it causes extreme direct and indirect medical costs. And the prevalence of allergy is increasing. So we believe that this is a large market and a true unmet medical need.
And I could add, just because I have a severe food allergy, that this is an unsolved problem. Currently, with my milk allergy, there are no treatment options available. There's no FDA-approved product, and so what I have to do is try my best. And this is the strategy that's given to millions of people who have allergies, is the prescription rather, is try to avoid what you're allergic to. And I can tell from personal experience that just doesn't work. For me with milk, there's just countless things that have butter. There's butter on the grill. There's milk and lime tortilla chips, hot dogs, rolls, guacamole. I've been accidentally exposed to allergens countless times at this point. And so I'm personally motivated, as well as everyone else on the team, because relatives and family members are affected, to really solve this problem.
Right, and epinephrine as a rescue therapy is great as an emergent rescue therapy if you need it, but it's not a solution, right? It's there as a last case resort, last case scenario if you have an acute reaction. But nobody wants to take their EpiPen. So we're trying to develop a therapy that people can take, have on board, and then not worry if they get accidentally exposed, which happens all the time. If you look at the literature, about 70% of ER visits and anaphylactic reactions are due to accidental exposure, meaning people were trying their best to avoid but had cross-contamination or something like that. So the other thing that I just wanted to touch on is that this is a worldwide problem. Globally, about 8% of the population may suffer from food allergies, and it's increasing across the globe. And it's incredibly prevalent in the US, in Australia, increasing in China. So, some people say that this is the next global pandemic is food allergy. But again, I want to stress that food allergy is our first target. We believe that IgE is implicated in a variety of disease, and one of the things that we've been really excited about in our research and discovery is we now have these reagents, these monoclonal IgE antibodies that no one has ever had before, and so we really believe that unlocks a lot of potential in drug discovery and development. So I'm going to turn it over to Derek now.
So now I'll provide some background on what the allergic cascade really is. What happens when someone is accidentally exposed to an allergen? So first, some background on the cells involved. We have two cell types that mediate the allergic cascade. One is called a basophil, and the other is called a mast cell. Each of these cells are granulocytes, meaning they're filled with these kind of granules or capsules that contain chemical mediators that affect some other part of the body. And so what happens is these cells are loaded on their surface with an antibody class called IgE. These IgE antibodies are bound to a high affinity receptor known as the Fc epsilon receptor, and there these cells wait. So basophils circulate in the blood, and mast cells are in tissues, and they wait until someone's accidentally exposed. What happens during that accidental exposure is someone, for example, me, accidentally consumes butter. So I'll, ingest butter, and there'll be some proteins that get absorbed into my bloodstream, for example. And that's what's depicted here by this orange triangle. That's an allergen, and almost all allergens are proteins. So that's an allergen coming into contact with an IgE antibody. These IgE antibodies are specific to these allergenic proteins. And so when these IgE antibodies bind said proteins, they cross-link the cells. And what this really means is that they initiate a signaling cascade that results in the degranulation of these cells. Degranulation is really just a fancy word for the release of the granules that are held inside those cells. And so what happens is, all of a sudden, all of these chemical mediators under the bloodstream and under the tissue, these cause all of the classic symptoms of an allergic reaction, from vomiting to diarrhea, hives, rashes, and ultimately, in very severe cases, anaphylaxis, characterized by, in most severe cases, difficulty breathing and ultimately shock.
Yeah. Exactly. And in the worst case scenario, death.
Absolutely, yes. And so what we seek to do is block the entirety of that allergic cascade. So what IgGenix is developing are IgG4 antibodies that have specificity to the allergens that cause allergic reactivity, but prevent these cells from degranulating. And they prevent cells from degranulating by binding and effectively outcompeting or blocking the interaction of IgE with those allergens. This mechanism of action of using IgG antibodies to block allergens actually has a lot of precedent and a lot of history behind it. This whole idea of a blocking antibody first came about in the early 20th century, where people were looking at pollen allergens and noticed that there was some compound in the sera that was able to bind or block pollen allergens. There's been a lot of literature since. I'll just go through some examples, but this is of course not a comprehensive list. In the 1970s, there were these really fascinating experiments where they took the serum of beekeepers who were tolerant to bee venom and passively transferred their sera to individuals allergic to bee venom. And what this passive transfer did is it provided protection from bee sting challenge in allergic individuals. So that told us that there was something in the serum, specific protein, that provided protection for beekeepers that could be passively transferred to allergic individuals. In 2011, there was a nice paper published on peanut oral immunotherapy. So it's probably a good time to pause and talk about oral immunotherapy, because it is one type of treatment that's being investigated to a greater and greater degree these days. Oral immunotherapy is a form of allergen-specific immunotherapy whereby someone ingests what they're allergic to in slowly increasing doses over time with the hopes of becoming desensitized. As you can imagine, because you're still allergic during the course of treatment, this can have a lot of adverse events, a lot of side effects that are treatment related. And ultimately, because it's attempting to modulate the immune system, this can take months, if not years, in people who it ends up being effective for, which is not necessarily everyone. So some challenges there, but despite that, people who undergo oral immunotherapy in some cases are able to reach a state where they're able to consume some amount of the food. And this is typically measured in what's known as an oral food challenge. So here, under clinical supervision, someone is challenged or is asked to eat in a blinded fashion what they're allergic to until they react or not. So this is the standard clinical endpoint that measures and is really the gold standard of diagnosing allergy. And so what you can see in this figure in the upper left is that over time, over the course of peanut-specific oral immunotherapy, the amount of IgG4 specific to peanut increases enormously. It increases by orders of magnitude over time, and this isn't seen in the placebo group. So there's something about the administration or oral ingestion of peanut over time that induces the immune system to produce high amounts of peanut-specific IgG4. So our thinking at IgGenix is, why wait the months and undergo all of these severe, adverse events when we can engineer an optimized IgG4 antibody and provide that with a subcutaneous injection that'll start to be effective on day one with no adverse events because we're not administering the allergen. So this was really, this peanut oral immunotherapy paper and others related to it was one of the kind of major sparks for this idea of why don't we provide an optimal form of the IgG4 antibody. Regeneron pursued such an approach, and they published a paper in 2018 in which they subcutaneously delivered a pair of monoclonal IgG4 antibodies to individuals that were allergic to cat. So they designed their antibodies, a pair of them again, specific to what is known as an immunodominant allergen within cat. So immunodominant just means that there are some proteins that drive reactions more than others. And for cat, FeLD1 is just one of those allergens that appears to be driving clinical reactivity more than the host of other proteins that are in cat dander. And so what Regeneron showed in this paper is that when you subsequently challenge individuals with cat dander, what happens is that these antibodies reduce clinical symptoms in that challenged environment. So this is measured by a total nasal symptom score, and as you can see in the lower right, that's significantly reduced all the way out to day 85 with a single injection. So a really powerful proof of concept for our mechanism of action in the field of allergy. Now that was for cat, and we think there are important differences for food, and we'll get into how our platform, we feel, produces better antibodies for such a treatment as we go further along. And then the last thing, and this was a very recent publication by a group in Switzerland, is one in which they used subcutaneous immunotherapy serum and showed that IgG antibodies specifically within that serum are responsible for the inhibitory activity in a mast cell activation test. So a mast cell activation test is exactly as it sounds. You take mast cells, you sensitize them, and then stimulate them directly with an allergen. So you're basically mimicking an allergic reaction in a test tube or in a plate of a well. And what they showed is that if you deplete that IgG, you're able to completely abrogate the protection provided by serum of individuals who have undergone another form of immunotherapy, subcutaneous, over the course of several years. So in summary, really a lot of precedent and a lot of literature that pointed us to this idea that we can design optimal IgG4 antibodies for the protection against allergic reactivity in individuals with food allergies. Here is the summary of our approach at a really high level. We are using single-cell transcriptomics, and I'll get into this soon, to isolate the extremely rare IgE-producing B cells from individuals with severe allergy. What this enables us to do is, using single-cell RNA sequencing, recover the full-length paired heavy and light chain sequences that comprise monoclonal IgE antibodies. And that's important because what we're effectively doing is discovering the very antibodies that are causing disease in allergic individuals. The last step for us is taking these IgE antibodies and re-engineering them as IgG4 in order to treat allergies. And we'll get into this reengineering process as well, but effectively what we're doing is we're taking the arms of the antibody you see in light blue here, which are the variable regions that bind to the allergen. We're retaining those and effectively grafting them on to a backbone that is an IgG4. And this grafting, or this reengineering or class switching, it's called, produces an antibody that retains its ability to bind to the allergen, but is no longer capable of eliciting the allergic cascade, and as we just talked about, actually affords protection in humans. I'll now talk about what we're calling our discovery engine, the seq sifter platform, and this is how we discover these IgE antibodies from severely allergic individuals. So first, some background on the IgE antibody class. I described how IgE is loaded onto the surface of mast cells and basophils via a high affinity receptor. But I'll add that it's actually still, even in allergic individuals, the rarest class of antibody in humans. It's actually orders of magnitude less abundant than more common isotypes like IgG and IgA. Just for reference, when you get a vaccine, the ideal response is a high production of IgG antibodies that will neutralize or opsonize that virus. IgA is involved in mucosal immunity, so microbiome and other barrier surfaces. You'll find IgA that affords you protection against a whole host of microbial community members. And then IgE originally evolved actually to protect us against parasitic worms. And this is where, part of the story around what gives rise to this increasing prevalence of allergies. Well, part of it, we think, the field thinks in general, that it partially has to do with the fact that our environment today is a lot different than our environment was in prehistoric times. We no longer are kind of under threat from parasitic worms. And so has the IgE antibody system, not just the IgE antibodies, but all of the effector cells and the cytokines that induce the production of IgE, have they been effectively redirected towards an otherwise innocuous target or innocuous antigen like peanut? And generally the thought is yes, but it's of course more complicated than just we're not exposed to parasitic worms as much anymore. Not only is IgE extremely rare in the serum, but the cells that produce these IgE, and these are B cells, are similarly extremely rare. There have been many publications throughout the years describing how difficult the challenge it was to actually isolate these IgE producing B cells from human allergic individuals. And therefore, it's been a challenge to discover the very antibodies that are causing allergic disease in humans. This was actually an unsolved problem for quite some time. There were very nice papers published in 2015, one by a group at Stanford and one by a group at Harvard, that isolated allergen-specific antibodies from allergic individuals, but these were all of the IgG and IgA antibody class. So it moved the field forward in that there were some then kind of descriptions of reagents and antibodies that might be involved in allergy, but still no one had isolated the very antibodies that actually cause disease, those of the IgE antibody class. So it wasn't until 2018 when one of our SAB members, Scott Smith, and myself published a pair of papers that were the first description of allergen-specific IgE antibodies from humans. And these were specific to fungal antigens and, in our case, peanut antigens. And this was a paper that came out in Science in 2018. In August of 2019, IgGenix was founded. Really, the idea here was that we have this platform, this novel platform, to discover IgE antibodies. And that was really great for moving the field of allergy forward in terms of characterizing the allergic response and better understanding the allergens involved. But we thought we could take it one step further based on all of the precedent I just described. We can take this from just the discovery platform to a therapeutic engine, one in which we discovered IgE and then re-engineered them to be blocking antibodies and treat allergies. So that was the idea. We were founded in August of 2019 and have since spent an enormous amount of time really industrializing what was an academic platform. This is a platform I developed at Stanford. It was very manual, very slow. It was very sophisticated in that it required a lot of really expensive research-grade equipment. And so we've since made a lot of modifications and optimizations to the program that allow us, with high throughput, higher sensitivity, better recovery, better specificity, to isolate these IgE producing B cells and ultimately recover the IgE antibodies they produce. So we're nearing now 10,000 unique monoclonal IgE antibody sequences, and these are from donors all across the US. So what Jessica set up was an IRB approved observational clinical study in which we recruited allergy clinics across the US. And so what clinical coordinators at these do is look for the most severely allergic individuals with, for example, clinical histories of high skin prick wheal test diameters, high IgE titers, previous episodes of anaphylaxis, visits to the emergency room, and from there are able to consent these individuals, and we get blood such that we can perform our discovery process and isolate the antibodies that are causing diseases for them. We focused a lot on peanut, as we'll talk about a little bit, but really we've been interested in applying this discovery platform to all sorts of allergies. So how does this work? This is a figure from our 2018 science paper. As I mentioned, we've changed it a bit since then, but largely it is the same set of overall steps. Previous methods were, really challenging or unsuccessful, and that's where single-cell RNA sequencing came in. When I initially had the idea or desire to build this platform, it was a number of years ago in which single-cell RNA sequencing was just taking off, and people were just starting to increase the throughput from literally single-digit numbers of B cells or any cells isolated to the dozens or hundreds. And so myself with an allergy, I was looking in the allergy field and realized isolating these IgE producing B cells was really an unsolved problem, and one in which single-cell RNA sequencing could be a really good fit. So it took literally years of failure to isolate any of these cells, and iterative optimization of every step from how the blood was collected in what specific tube to how it was handled, processed, enriched, any depletion steps, the single-cell sort scheme, and then all of the single-cell RNA sequencing process in order to yield a full pipeline where we go from blood to ultimately IgE antibody sequence. So single-cell RNA sequencing is quite powerful, and I'll talk about why single-cell on the next slide. But it's really a technique that gives you a lot of insight, not just into the antibody sequence, which is for us the most important component, but it also tells you a lot about allergic disease. It tells you about the B cell subtype of the B cell that's producing these IgE antibodies. So it gives you gene expression. It tells you about activation state. You could look at splicing and the expression of, for example, membrane-bound antibody. And so it gives you a wealth of information that you can couple with the antibody sequence and ultimately antibody specificity to gain a more comprehensive understanding of allergy. Once we have that antibody sequence, and this is through a bioinformatic process known as assembly, where we go from short next generation sequencing reads to full-length paired heavy and light chain sequences, what we then do is we re-engineer, as I mentioned, and then clone and express those antibodies such that we then have the physical protein in our hands and can perform all sorts of specificity screening and affinity assays. Now, as I mentioned, single-cell RNA sequencing is really the foundation of this platform. Why study single cells at all? Why can't we just get a blood sample, lyse the B cells, and then use RNA sequencing on the mRNA released from those cells? Well, it really comes down to the structure of an antibody. An antibody is a heterodimer, and it's comprised of 2 identical heavy chains. They're in purple on the left, and 2 identical light chains in green. And the important part is that these 2 chains are encoded by genes on separate chromosomes. So what happens is if you lyse those cells, you lose the critical pairing information that dictates the function and specificity of the antibody. And that kind of bulk loss of pairing is shown on the right here, where you have cell 1, purple heavy chain and a green light chain. You have cell 2 with a blue heavy chain and an orange light chain. What happens when you lyse those cells is you've mixed the mRNA of all of those transcripts, and you can, even if you sequence it, no longer recover the critical pairing information of purple and green and blue and orange. So that's why we really, if we're interested in antibodies, have to study single cells. Another one step deeper in kind of the single cell RNA sequencing process is illustrated in this slide. As I mentioned, we start with blood from severely allergic individuals, and our isolation step involves fluorescent activated cell sorting individual B cells into individual wells of a 384 well plate. Once those cells are sorted, we lyse those cells, and that releases the mRNA into the well. We reverse transcribe that mRNA, and there's some molecular biology tricks here that then allow us to amplify the entire transcriptome, so all of the mRNA in the cell, with a single primer. And this is because in the reverse transcription step, you perform basically a segment switching process, a template switching process that adds a primer binding site to the 5' of the mRNA molecule. In other words, we're able to just capture all of the genes the cell is expressing with a single primer during PCR amplification. This is powerful because the genes the cell is expressing includes our genes or transcripts of interest, the heavy and light chain sequences of an IgE antibody. The last step for us is library prep. And so in this case, what we're doing is we're adding nucleotide barcodes to the single cell cDNA of each well. And so we add this as a unique 8 or 10 nucleotide sequence that allows us to distinguish from which cell the reads came from in next generation sequencing. The next generation sequencing step is shown there on the right. This involves pooling all of the cDNA and then reading it out on standard Illumina sequencing instruments like the NextSeq or a MiSeq or a NovaSeq. And then lastly, the data is analyzed. I've tried to present here kind of a general overview of the steps involved in single cell RNA sequencing that isn't necessarily specific to plate-based single cell RNA sequencing, which is what we're using. There are a lot of new approaches, new companies, and new ways of doing single cell RNA sequencing that makes the field more accessible to those that are not as familiar with it or not ready to make the capital investment in performing single cell RNA sequencing. But for us, we use plates, and when we process those plates, what's really important is liquid handling and throughput. We source a lot of blood, sort a lot of cells, and that means we need to process a lot of 384 well plates through the single cell RNA sequencing process of reverse transcription to amplification to library prep, and there's others involved that I'm skipping over for the sake of brevity. And what really helps us here is liquid handling instruments from SPT LabTech. We are very happy users of the Dragonfly shown in the center there, and the Mosquito high volume shown on the right there. And these two instruments really pair nicely in allowing us to respectively distribute buffers or master mixes to all wells of the plate, and then perform sequential column-wise operations on those plates, be it transfers or poolings or mixing. So it's been really nice and has accelerated the progress of the company having liquid handling in-house. And another benefit actually I'll add is the ability to miniaturize. the biotech climate is not great at the moment, obviously, and so we cherish every opportunity to save on reagent costs, and the SPT LabTech instruments have allowed us to do that. We've miniaturized a number of the assays by anywhere from 5 to 20-fold. And this is really translated to a direct savings in reagents.
And time.
And time. We think, yeah, this is a really powerful way to save on both time and cost, and reduce errors. So 3D 4-well plates are, if anyone's ever handled them, very difficult to handle manually and accurately. What's great is that you can set a protocol for any plate size for the dragonfly, and for us, the 3 to 4-well plates from the mosquito, and have it perform nearly flawless kind of operation of whatever protocol you want, be it just simple transfers. You're doing qPCR, for example, then you move some amount of cDNA to a new qPCR plate, or more complex protocol steps like library prep, in which we have 4 different plates on the deck corresponding to either cDNA or reagents or a destination plate and barcoding plate. So it's really kind of amenable to all different workflows, and we're expanding its use actually to other parts of the company, not just in discovery. So now, after this step, we have our IgE antibody sequences. So we've discovered the sequence. We have the variable region, the heavy and light chain variable regions that belong to these IgE antibodies. The next step is really taking those and class switching them or re-engineering them such that the antibodies we produce are IgGs. So that involves starting with a pair of plasmids, one for the heavy chain, one for the light chain, synthesizing the heavy and light chain variable regions, cloning those into the respective plasmids. Those plasmids are then co-transfected into CHO cells. Those CHO cells secrete those antibodies into the supernatant. The supernatants then pass through a protein A column to collect the IgGs, and then the antibodies are subsequently eluted. And so I've provided some kind of rough timelines here for how long these steps kind of typically take. But you know, for precious samples or you know, really important kind of high priority work, all of these can be accelerated and the timelines decrease. After this purification process, we have in our hands an IgG antibody that has IgE variable regions. And so for us, we actually don't know the specificity of the antibody at this point. And this is best illustrated by an example. Because our discovery platform is unbiased and we're isolating all of the IgE producing B cells, if someone is allergic to multiple things, those IgE antibodies, and then re-engineered as IgG, could be specific to multiple allergens. So we have one donor where we've received a number of blood samples from them, and each time we've isolated antibodies to shellfish and antibodies to cat, because this donor is allergic to both cat and shellfish. So for us, we have a step of IgG antibody characterization, so effectively specificity screening. We're actually starting to use the SPT LabTech instruments in these types of protocols as well, just due to the ability to screen at higher throughput more molecules against more allergens. At a high level, I've gone through really the first 2 steps in what we consider a 3-step paradigm. So the first is discover. We use single-cell RNA sequencing to discover IgE antibodies. We build. So this is re-engineering IgEs from humans into IgG4s that might offer protection as therapeutics, and then validate. So this is something we haven't talked about, but we'll touch briefly at the end. This is where we want to understand not only how well these antibodies bind to an allergen, but can they block allergic reactivity in vitro, ex vivo, and in vivo. So we'll run assays like one I mentioned earlier, the mast cell activation test, in which you have IgE antibodies sensitized, or you have mast cells sensitized with human IgE. You stimulate those cells with allergen, and then you measure in the presence or absence of our IgG antibodies whether we're able to inhibit that cellular activation. There's a similar assay known as a basophil activation test. Here is an assay where we get fresh whole blood directly from allergic individuals, and you can stimulate the blood sample and the basophils within that sample with allergen, and like the mat, do so in the presence and absence of our IgG antibodies, and therefore measure the inhibition in this really translational assay of cells directly from allergic individuals. And then lastly, what we do in vivo is through collaborations work with animal models where the animals are orally sensitized to an allergen and then challenged orally with that allergen. And we measure whether our antibodies provided beforehand subcutaneously are able to prevent anaphylaxis in those animals. So those are really the three steps: discover, build, and validate. And overall, we think this approach has a lot of advantages. One is that we're leveraging the affinity maturation process performed in the immune system of humans with allergies. So I myself have been allergic my whole life and been accidentally exposed countless times. And this has given my immune system, as the immune system should work, the ability to kind of refine its response against what it deems to be foreign or undesired. So my immune system has affinity matured, really high affinity antibodies. They're able to bind really tightly to these allergens and trigger the allergic cascade upon kind of very small amounts of allergen exposure for me. Unfortunately, we believe that our discovery platform is great and that these antibodies are coming from humans and will end up in humans. By staying kind of in human the whole process, there won't be as many kind of immunogenicity concerns as compared to other technologies where you might source antibodies from another species or through kind of random recombination like phage. The most important advantage that we feel is that our IgE antibodies discovered in an unbiased way naturally tell us the most important allergens and the most important epitopes on those allergens that we need to block in order to develop an efficacious therapeutic. Here's where we are as a company at the moment. As I mentioned, we have nearly 10,000 unique monoclonal IgE antibody sequences. These have specificities to effectively everything humans are allergic to. So we have antibodies against all of the major food allergens: peanuts, tree nuts, shellfish, fish, egg, milk, soy, wheat, etc. We also recently discovered antibodies against alpha-gal, which is a sugar on certain mammalian proteins that causes what's known as a red meat allergy, something of increasing prevalence in various areas of the world, but very commonly in the southeastern United States. We also have antibodies against all sorts of non-food allergens. This is really a benefit of the unbiased approach we've been taking for discovery. We've been recruiting from allergy clinics, as I mentioned, individuals with food allergies, but oftentimes these individuals would be also allergic to other non-foods. And so we'll discover kind of accidentally or as a byproduct of our process, antibodies specific to pet danders like cat and dog, as well as pollens from grasses, trees, and weeds, as well as molds and even a few to parasitic worms. So I'll present kind of two slides on what's next for IgGenix as a company. And this by way of kind of our progress in peanut allergy. So first will be just some overview on the animal models themselves, and then I'll get into how we measure the efficacy of our antibodies. So first, to start off on the left, what you can see is the sensitization protocol. So this is the amount of work that goes into getting mice to a state where they're allergic enough to be challenged and have a measurable response to that challenge. And so the challenge is with peanut provided intragastrically. So it's basically oral administration of peanut to mice that have been sensitized over the course of 2 months. And what happens when you challenge these mice after 63 days is you see a swift drop in temperature. So that's the temperature plot shown on the right. Two curves, blue and green, correspond to saline, so that's a control, as well as a control antibody trastuzumab. This is irrelevant for allergy, and so should offer no protection for the mice. Indeed, that's what you see. At 15 minutes after providing this peanut extract, you see a swift drop in body temperature for these mice. But when we provide our antibodies on day 61, 2 days before the challenge, what you can see is that these antibodies offer protection against this oral challenge and prevent anaphylaxis in a dose-dependent manner in these animals. So,
and I just wanted to interject, in the field of allergy and immunology, this type of test, oral sensitization and then an oral challenge, is thought to be most relevant and clinically translational. And indeed, in oral immunotherapy with peanut, and again, that FDA approved product for peanut, they showed these types of studies and this type of response.
Absolutely. So we're really confident in the translatability of this model, given it's with kind of full peanut extract, and these animals have been sensitized orally with peanut. As you can see by the gray, purple, and orange curves, our antibodies in a dose-dependent fashion protect against anaphylaxis. And so we're really excited by these results, especially considering the dose of peanut that these animals are given in order to elicit this response. So it roughly correlates to about 300 peanuts in a 70-kilogram human, which is an enormous amount that someone with an allergy would never be consuming. So it just speaks to the potential of our antibodies to effectively sop up an enormous amount of allergen that gets in through the gut. So we're really excited about this, moving our peanut program forward, and eventually getting to the clinic where we can evaluate whether these antibodies can help human allergic individuals who've been suffering from allergies without great treatment options. So I'll end here really on kind of what IgGenix is thinking about in terms of not just being a therapeutics company, but ultimately building an understanding, a more complete understanding of IgE biology and allergic disease. And we really think it comes from integrating all of these facets of allergy, all of these different modes of understanding, perhaps the donor or diagnostic information about that blood sample with expertise in protein sciences. So we've invested heavily in being able to model the structure and the binding of our antibodies to allergens to actually express the allergens recombinantly. So we have reagents to work with in order to measure affinity, measure specificity, map our antibodies onto the allergens, and then also on the protein side, understanding the relationships of antibodies within an individual over time if we collect repeated blood samples, or I would say more importantly, understand the relationships of antibodies between individuals. We've seen a number of instances of this really interesting phenomenon known as convergent evolution, whereby totally unrelated individuals have recombined nearly identical antibodies that target immunodominant epitopes on immunodominant proteins. And part of that is, then actually doing the bench work to confirm the expected specificity, confirm that these antibodies are high affinity like we want for an efficacious therapeutic. And then we're thinking next steps in terms of innovation. We've talked about a blocking IgG antibody, but that's really the foundation and the first step. We're also envisioning being able to design novel constructs that have additional functionality or behavior in the immune system, inhibitory mechanisms of action, so not just stoichiometrically outcompete endogenous IgE for allergen, but actually actively inhibit it or suppress the cells involved in allergic reactions. So why don't we end there and start to take questions from the audience.
And please follow us on LinkedIn and Twitter. We're there for you. And you can contact us at info@IgGenix.com. Those emails actually come to me, so I'll respond. So Megan, any questions from the audience?
Hi, yes. Thank you very much to both of you for those great presentations. Before I go on to the questions, I'd just like to remind everyone that this webinar will be available to access on demand on the DDW website, which is www.ddw-online.com. So now I'll move on to the questions. Firstly, Derek, it sounds like you are well underway to address allergies that affect so many people day to day. What does IgGenix need to take the next steps you described?
So we're actively taking those steps now. For us at a high level, what comes next is once we decide on the antibodies we're going to use for a therapeutic, we initiate work with a contract development and manufacturing organization, the CDMO. And the CDMO, what they do is they effectively produce at a high scale with high purity under FDA regulations, a GMP product that will go into our phase 1 clinical study.
But yeah, I'd also add to that, doing drug development is expensive, and we're actively in the process of raising our next round of financing. So if anyone out there knows investors who are interested, please do contact us, because we're just in the process of raising that next round.
Fantastic. So now the next question I have here is, what are the advantages of your discovery platform over conventional antibody discovery formats based on either immunization of animals or phage display against purified targets?
Yeah, great question. There are a lot of approaches to antibody discovery, and each has its advantages and disadvantages. Animal immunization has been kind of a bedrock of the biotechnology industry for a long time. Here, the process involves taking your antigen of interest that you want antibodies against and injecting that into an animal kind of repeatedly in order to elicit an immune response in that animal that you then harness for a therapeutic. Some advantages here is just kind of time and how well it's worked historically. Some of the disadvantages might be that depending on the specific animal or strain, you might have to do what's known as humanization after that. You might have to basically make the mouse antibodies or rabbit antibodies look more human, so to speak, in sequence space. There's newer animals that have a partial human immune system that avoid that step. But the challenge still remains is that you don't know necessarily in the case of us for allergies that the antigens and the epitopes on those antigens being targeted by the animal is the same as that which is targeted by the human immune response. So that's why we feel with our platform, which is fully human, we're isolating the antibodies causing disease. Those are naturally, by definition, the ones that we need to block in order to have an efficacious therapeutic. And as I mentioned earlier, compared to phage display, we expect fewer immunogenicity concerns because these antibodies were being produced in otherwise healthy allergic individuals.
Thank you. That was a great answer. The next question I have here for you is, how widely applicable is this technology? Could it be utilized to develop therapies against other allergies like seasonal hay fever?
Absolutely.
Yeah, absolutely. We perform an unbiased discovery process, and so we have antibodies against all sorts of seasonal allergens. And we found some, really interesting antibodies in terms of their cross-reactive properties. So antibodies that are binding not one or two, but a whole host of different grass, tree, and weed species. So absolutely, it's applicable. It's just, given our small team of about 11 people in South San Francisco, difficult to pursue everything we're interested in all at once. And so it's also probably a good time to add we're interested in partnerships on any of these other programs, especially seasonal or pet related.
Yeah, and the other thing that I would say is we believe that allergens in general are just the tip of the iceberg. and that we can see IgEs are implicated in a whole host of diseases. And in fact, we have discovered IgE to non-allergens, to parasites, some viruses, and sort of the list goes on. So, you know, we definitely think that allergy is the low-hanging fruit here, but there's many areas to be scientifically integrated in the future.
And I would say absolutely, because we now finally have these IgE antibodies, which are effectively reagents for understanding allergic conditions better.
Great, thank you. Now, the last question I have for you here is, how much protection do you expect the therapeutic IgEs to confer? Enough to protect against accidental exposure in small doses, or could a patient consume peanut products safely after treatment?
So I'm going to take that one. That's a great question. So the honest answer is we're not in humans yet, so we don't know. As Derek showed from our mouse studies, it appears that those mice can ingest quite a bit of peanut. But what we know about the FDA and what they approved for the one approved peanut allergy product, the phase 3 endpoint was being able to tolerate a 600 milligrams of peanut protein, which is about 2 to 4 peanuts. And what that's called in the field is being bite safe, so that people are protected against accidental exposure. So we believe that will be our FDA endpoint as well. If you talk to patients and caregivers, they really don't want to eat the food. They've sort of grown up and they're averse to the food, but they do want to be protected against accidental exposure. So that's certainly going to be our goal. If we can get beyond that and provide more exposure protection, we'd love to do that as well.
Thank you to both of you for such detailed responses. There's no more questions from my side. And so for our audience, I'd just like to say that you will find SBT LabTech's handling guide in the handout section, which you can download to learn more about making the move to automated liquid handling. And if you'd like to listen to this webinar again, it will be available on www.ddw-online.com. Thanks for listening, and thanks again, Jessica and Derek.
Thank you.
Thank you.